Okay, we left off talking about how temperatures affected can affect protein structure, and I guess it might be worth explicitly pointing out what is going on there. So if we have a three-dimensional structure of some protein, that three-dimensional structure is maintained by weak chemical bonds that we've already covered. For example, parts of the structure that are alpha helices are held together by main chain atoms engaged in hydrogen bonding. Likewise, there can be hydrogen bonds between certain amino acid side chains and other amino acid side chains. There can be hydrogen bonding there. In addition, there can be salt bridges between positively charged amino acid side chains and negatively charged amino acid side chains, the basic side chains and the acidic side chains that are found on different amino acids. And what increasing the temperature does is it increases the kinetic energy of molecules in solution that bombard this protein as they hit it. Molecules are moving in solution and their kinetic energy rises with increasing temperature to the point at which, at some point above a certain temperature, they will cause the breakage of weak bonds that are holding together protein structure, either weak hydrogen bonds or possibly the salt bridges or the ionic bonding between positively charged and negatively charged side chains. And the protein then begins to unfold. And as the temperature increases further, you can actually get full denaturation. So increasing temperature at high temperatures can be detrimental to protein three-dimensional shape and can disrupt protein function for that reason. But we pointed out that there's a temperature optimum for most proteins. And this is activity of the enzyme, and this is temperature. So we see that raising temperature above a certain point leads to changes in three-dimensional shape, which can be detrimental to function. But why is this increasing at lower temperatures up to the optimum? Well, that is simply increasing the kinetic energy of molecules in solution so that molecules can find the active site of enzymes and be acted upon by enzymes. So enzymes actually function better as temperature increases up to their optimal point, at which point changes in three-dimensional structure can lower enzyme activity. Now let's move on to another, one more thing I want to point out, and that is that different enzymes have different temperature optima. So if this is activity, if this is temperature, we might have one enzyme that has, for example, one of your normal enzymes functions optimally at 37 degrees centigrade. But if you were to take an enzyme from a bacterium that lives in a hot springs, let's say in Yellowstone Park, it might have a temperature optimum of 72 degrees centigrade. So depending on the structure of an enzyme or a protein, its optimum may be quite different depending on what environment the organism that holds that enzyme has evolved in. Now another environmental factor that can influence enzyme activity by influencing enzyme structure is pH. So if we look at pH down here, we know that most enzymes prefer pH values from 6 to 8 near the neutral pH, but some enzymes function optimally at very drastically different pHs. For example, the digestive enzymes in your stomach function optimally at a pH of 2, the very acidic environment in your stomach. So just like temperature, if we look at the activity of a particular enzyme versus pH, you will also find that most enzymes have an optimal pH. Some enzymes have different optimal pHs. And that optimal pH, of course, is where activity is highest. Now, how can pH, the concentration of hydrogen ions, influence the activity of enzymes? Well, remember, in proteins, there are acidic amino acids that have side chains that sometimes have an acidic group, a carboxyl acidic group on them, where this oxygen can become negatively charged if it donates a hydrogen to solution, or alternatively can become neutralized if it accepts a hydrogen ion from solution. So here the hydrogen ion it bonds to this oxygen. So here we have an uncharged acidic side group of an amino acid, and up here it is charged by virtue of donating a hydrogen ion to solution. Well, how can hydrogen ion concentration in solution then, the pH, affect this? Well, at very high, so if we look at, let's look at it this way, COO, let's call it COO minus, indicating this carboxyl group, and plus hydrogen ions yields COOH, uncharged. So this is just a little reaction here. Now at very high concentrations of hydrogen ions, as hydrogen ions concentration increases, that will drive the reaction to the right because there's lots of reactant. And so at high hydrogen ion concentrations, that is low pHs, and at low pHs, acidic side groups will become uncharged. Whereas at higher pHs, when the hydrogen ion concentration is very low, the equilibrium will be shifted to the left, and this hydrogen ion, this carboxyl group will ionize, producing free hydrogen ions, donating them to solution, and putting a negative charge on that oxygen. So pH can affect the charge of acidic side groups on proteins, on amino acid side chains in proteins, like enzymes. And likewise, there are side chains of amino acids that are basic amino acids that have amino groups on them. And those amino groups can accept hydrogens from solution. When that happens, they accept a hydrogen and they become positively charged. Alternatively, when the nitrogens have donated a hydrogen to solution, or when they're not protonated, they are uncharged. So again, you can say an amino side chain plus hydrogen yields NH3 plus in this case. There's our charge when protonation occurs. Now, if hydrogen ion concentrations are very high at acidic pHs, in other words, that will drive this reaction to the right rather. High concentration of hydrogen, low pH, this will become charged. This amino group on a side chain of basic amino acids will become charged. Whereas at low hydrogen ion concentrations, NH3 will donate hydrogens to solution and become uncharged. So think of a protein now which has ionic salt bridges or ionic bonds between positively charged side chains and negatively charged side chains. Say positive, negative. These are ionic bonds between side chains of amino acids that help stabilize a protein's three-dimensional structure. And then you can easily see how affecting the charges on acidic and basic side chains by changing pHs can affect these salt bridges and disrupt protein function. That's why there is an optimum pH usually for a protein, below which it does not function optimally because its three-dimensional shape is altered due to protonation or deprotonation of particular side chains, and likewise above that optimal pH, a tertiary three-dimensional structure is disrupted by disrupting these bonds caused by affecting the charges on acidic or basic amino acid side chains. All right, so this is shown a little more neatly here. We have a human enzyme versus a hot springs prokaryote. You can see the optimal temperature for the hot springs prokaryote is much higher than that for a human. And here we have an optimum pH for trypsin at 6.5, but for pepsin it's down about 2.5. So again, optimal pHs based on the effect of pH on three-dimensional structure. Now enzymes, the catalysts that run biochemical activities, that run biochemical reactions, can be affected by inhibitors. And there are two types of inhibitors, competitive inhibitors, and a competitive inhibitor would compete with the substrate for binding to the same active site. So if this is the active site of a protein, and here comes a substrate in here and binds to it, if instead an inhibitor comes and binds in here, then the substrate can't gain access to that active site. So naturally, a molecule which can mimic a substrate for an enzymatic reaction can bind to the active site of an enzyme and prevent the normal substrate from engaging the enzyme. Therefore, these competitive inhibitors inhibit enzyme function by blocking the active site. On the other hand, non-competitive inhibitors bind to sites we call allosteric sites that aren't the active site. So if this is an enzyme here and this is its active site right here, and this is an allosteric site where inhibitors can bind. Substrate can bind here, that's great, so the enzyme would like to do that. But if an inhibitor is bound at an allosteric second site on the enzyme, that can change the protein's shape such that a substrate cannot engage the active site anymore. So non-competitive inhibitors aren't competing with substrates for active sites. Rather, they're binding to second sites on an enzyme that can alter the enzyme's function and alter its ability to engage substrate. And so these so-called allosteric enzymes, because they can exist in either an active or an inactive state, depending upon whether an allosteric molecule has bound to a second site on the enzyme, we call those allosteric enzymes. And their allosteric inhibitors then bind to an allosteric site that inactivates the enzyme. It alters the enzyme's three-dimensional shape and prevents it from acting appropriately. There are allosteric activators that bind to a second site, however, not the active site, but they bind to a second site and activate the protein, maybe change a shape that makes it more amenable to substrate binding. So you can have allosteric binding sites on enzymes are non-active site places where molecules that can affect enzyme function bind. Either the allosteric inhibitors or allosteric activators can bind there. So here's an example. Here's a competitive inhibitor. This green molecule here is binding to an active site of an enzyme, blocking competitively the binding of substrate there. So this is a competitive inhibitor. Whereas an allosteric inhibitor can bind to a second site on an enzyme, changing its three-dimensional structure in such a way that the substrate is no longer engaged. So this is a non-competitive inhibitor. Now, metabolism describes where we're going to be going next. We're going to be looking at the chemical reactions that occur in an organism. And particularly, we're going to be looking at anabolic metabolism and catabolic metabolism. And anabolic reactions are reactions that use energy to make new chemical bonds. So you can think of this as building up. If we look at metabolism, so if we look at metabolism and divide it into anabolic and catabolic metabolism, anabolic, we are building up compounds. We could call this biosynthesis, if you will. So we are building up substances, and this requires an input of energy. So energy is put into this system. For example, when plants use photosynthesis to build up sugars and starches and carbohydrates, they're using the energy from the sun to do that. And they're synthesizing organic molecules, sugars, that can be used for energy. On the other hand, catabolism involves the breaking of chemical bonds, releasing energy. So energy is released in catabolism. So when you burn food to derive energy from that, you are breaking down substances. You're breaking chemical bonds. So anabolic pathways and catabolic biochemical pathways are opposites in terms of metabolic pathways. Anabolism is building up compounds, biosynthesis, and catabolism is breaking down, releasing energy. And we're going to be considering these two types of metabolic pathways shortly. Now, some enzymes require additional molecules to function appropriately, and we can have cofactors that are metal ions that often participate in catalysis, and we'll see cofactors when we get to DNA synthesis. Coenzymes are non-protein organic molecules that can accept electrons in redox reactions or donate electrons in redox reactions. So these are two additional molecules that bind to proteins and to enzymes and affect their activity. The last part of this lecture will be very brief, and we'll just be talking about some features of biochemical pathways before moving on to specifics of anabolic and catabolic biochemical pathways.